So, in this neighbor joining tree method with the star decomposition, remember we're taking that and we're turning it into little groups of branches that we can then connect up. Just a different way of doing what UPMGA, yeah, UPGMA, whatever it is, was trying to do. So we are going to, specifically, we're going to pair things. So we're going to find least distant pairs, and so we're always going to group things in pairs, and then we can take that pair and group it with another thing, but we're still taking the pair as if it's one thing and grouping it with one other thing. So it's always like you're grouping two things. Yeah, you're just grouping the average of these two things to to another sequence. And there's your group right down there where they join together. My thumbs are touching at the node. Ah. And so at each stage, two terminal nodes, like two branches or two of the farthest nodes from the root, are going to be replaced by one new node. Not even necessarily replaced by, but we're going to add one that joins them together, really. And there's going to be a certain distance between those based on the genetic distance between the two nodes. Yeah. And by genetic, I guess I mean molecular distance, because it could be a protein distance or like a gene, DNA or RNA distance. Okay. So we're going to continue this process just like with the other one until we have joined every single thing together. And so once again, if you put in sequences that are not homologous, if you haven't checked that out already, then this algorithm will cluster them together without thinking twice about it. They will just have really long genetic distances. And if that makes no sense in terms of what we understand about the genes themselves that you are clustering or proteins, then your tree will also make no sense. Yeah. So it's only as good as the information that you put in. And there's a big component of screening, like you have to find genuine homologs before you put them into a tree like this. Otherwise, you just get nonsense out of the tree. Okay. So neighbor joining algorithms are going to minimize the total number of branch lengths in order to make the tree. At each stage, as they're clustering, they're going to minimize the branch lengths to make those two join. Now, the final tree is not necessarily going to have the smallest overall branch lengths, but each step along the way, it had the smallest length to join those two nearest neighbors. It's going to be close, but it's slightly different. Yeah. And so maximum parsimony is going to be shortest tree. Neighbor joining is going to be clustering the things with the least difference between them. Just like the other method. Yeah, and neighbor joining trees are going to be unrooted trees. So let's go through an example. So here's a bunch of sequences. That's great. These are all neighbors. And even though these are all individual sequences, we will call them operational taxonomic units. Because we're not saying if they're all species, genera, if some are in a different family than others. We're just saying these are all some kind of unit. And we are going to join them together in the way that makes the most sense in terms of minimizing the distance between pairs and minimizing the distance between neighbors, basically. So there you go. Okay, step one, star-like tree. Voila. With a single interior node, which we're just arbitrarily defining. Yeah. But yeah, it's totally arbitrary. Step two, we're going to find the two closest ones based on our distance matrix. And we're going to connect those via an internal branch, and then we'll connect everything else by a second branch. So we started with node X, and so node X remains here. We're going to push everything else out on node Y, because the two on node X are closest to each other, and so that's fine. You can mentally swap these around if it makes more sense for you, and that's not a question on any test. This is just, can you kind of get your head around it kind of deal. And so then we're going to repeat the process until we join everything up. So there's our star-like tree. There's 1 and 2 joined together. All the rest separate. And then we're going to go to, let's see, 1, 2, and 3 joined together. And all the rest of these different. There is a node right here. That was our old X. And there is a node here. We're joining those together. Oh, here, sorry, we're going this way. Da, 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 and so on. Joining things up, joining the nearest neighbors, joining. And so we're basically taking a tree and then turning the tree, we're pruning the tree. We're merging branches of the tree to make thicker branches. It's like if you took a bunch of sticks and you glue some of them together to make the root of the tree, and a bunch for the branches, and then fewer for the little branches, and then the leaves are the little data points, like the ends of your sticks. That's kind of how I think of it. Whereas UPGMA is taking a bunch of little, think of them as like acorns on the ground, and drawing circles around them and saying, okay, these two are close, these others are close, and now these are closest, and until eventually you've got the entire thing grouped up in the way that makes the most sense on a two-dimensional plane, basically. Advantages: fast. Now the fun word here is bootstrap. And you can say, oh, it's like pulling yourself up by your bootstraps. BOOTSTRAP. Yeah. And it's not really that, but it kind of is. So the way you bootstrap a tree is you're basically going to run the tree over and over again with similar sets of data. And you're not going to take any one of those as correct, but you're going to take like the results of 100 different trees. You're going to see how they overlap, which branches are always there, which branches are sometimes different. And they say for these branches, you would say 100% support because they always line up. For these branches, you would say they are not supported 100% of the time. They're like, in 50% of the trees because I only have two hands. Yeah. But with 100 different trees and lots of different branches on them, yeah, you get nice nuanced branch support values or node support values, you could think of them as. So bootstrap analysis is when you do this. And so you would say, all right, that makes sense. I would very much like to do that, but I only have these 10 sequences. How do I do that? The answer is you use different parts of the sequence. So what if you made a tree using only the first, third, fifth, seventh, ninth, only the odd nucleotides in that? And you get a particular thing. What if you made a tree using only the even nucleotides? You get another tree. How close are they? If everything's homologous, they should be roughly equally close. It's a different way to use half the data and get kind of an independent answer. What if you use the first half versus the second half? What if you used, 1, 2, 5, 6, 8, and the other numbers, boom, and built a tree that way? Those would all be different ways to bootstrap. And so you can just slice your data into different bits, and the computer will do this for you. And then rerun the tree and then say, all right, out of the 100 trees, 1,000 trees, million trees I made, what percentage of the time does this particular branch come out with this topology? Remember, topology is basically which things are going to be clustered on that branch. Are these two always clustered together? Is there something else clustered on the branch? Yeah, that would be a different topology. But yeah, if the same things are always clustered in the same ways, then we would say that's the same topology. Now, topology is not going to have anything to do with genetic distance, like the length of the branches. It's only going to have to do with the way that the branches are connected to each other. So that's where we will pause for right now.